Okay, continuing genetics basics. So people had an understanding of how coding worked and how traits were produced. And they had this whole set of theories about evolution and natural selection, selecting for adaptations for traits, basically. But now they knew how the traits originated. Now, evolution works on populations, right? It changes the average member of a population by conditions change, and those conditions kind of select the winners from a population and the losers from the population based on the traits they have that fit the new conditions. For those of you who completely blew up on that question on the exam, right? Conditions change. Those with features that are good for the new conditions are more likely to survive, pass those on to offspring. They become more common generation after generation. The offspring is that they don't change and then pass the changes on. It is amazing how often biology students are Lamarck at heart and they still can't get it out of their head. I know it's terrible that we use the term adaptations, but no, is that they don't change and then pass the changes on. But now they knew where the traits came from. And they started to apply genetics in what's called the modern synthesis. What's a population? It's a collection of individuals. Except, no, it's not. It is a huge collection of alleles. Those individuals carry alleles for the same genes, but differences in the alleles produce differences in the traits. So what evolution is selecting for isn't the traits, it's the codes for the traits. And what the population then is it's a huge gene pool with alleles in different frequencies. And an allele that's relatively rare, but produces a trait useful for certain conditions. The conditions change in that direction. Now those alleles produce a benefit for the possessors, for the holders of those alleles. They're more likely to be the survivors. And so those alleles get passed on to the next generation in a much higher ratio than they started in the population. They become more common in the next generation and the generation after that, as long as the new conditions persist, is that the allele ratios shift over time. And so your gene pool shifts over time. What we used to consider the average individual is now a representation of what's the more or less common alleles. As conditions change, alleles that are useful for those new conditions become more common, ones that are not so good for those conditions become less common, and the gene pool shifts. Connecting into this in terms of genetics and evolution, we'll go back over this, we've had this a few times. Asexual reproducers make genetic copies, right? That's the definition, asexual reproducers, not reproduction without sex. It's the way that offspring relate to the originators. In this case, offspring are genetic copies. They get the same alleles that their parent had. And that is great for reproductive purposes. You're actually reproducing. But if conditions change and the conditions don't suit the parent, they don't suit the offspring either, because they're all genetically similar. But are they? Mutation rates are not high, as we'll talk about later. Most mutations don't do anything. Is that they don't change a protein, or they don't change a protein in a significant way, and so most mutations are neutral. If they do change what a protein does, they're much more likely to mess the protein up than to make it better. But if, as an asexual reproducer, you're cranking out huge numbers of offspring, those rare mutations that will work in a change of conditions, the numbers start to work for you. 1 in a billion chance when you've cranked out 10 billion offspring starts to work for you. So this applies to asexual reproducers, and the rate of mutations, which we just talked about, it requires huge numbers. Yes, you spread your offspring out. Often conditions are very local for asexual reproducers. They tend to be small, single cells. So they're in microenvironments, and it doesn't take much movement to get into a different microenvironment where conditions may not match. So those are the two advantages to cranking out lots of offspring to asexual reproducers. Is conditions change, instead of being basically screwed, maybe you spread your offspring beyond those conditions, or maybe by cranking out lots of them, you get a mistake that actually works for you, like makes you resistant to penicillin. For sexual reproduction, remember, it's terrible for reproduction. You're not actually making copies. That the offspring are a genetic mix of two sources, even from the same individual. If you've got a bunch of chromosomes, you're mixing the pairs differently, and your offspring are going to have differences from you in terms of, maybe you've got a dominant and recessive, and they get the two recessives, or they get two dominants. And all of the different mixes you can get. So it increases diversity. So when conditions change, your offspring are different enough that they've got a better chance of having beneficial mixes of traits in the new conditions. That's the upside of sexual reproduction. Downside is you're not making copies. You're not reproducing. Then you have alternation of generations. I think we've talked about this. I know we talked about it first semester. Now, in some organisms, you have what's basically a fudge factor. Bacteria produce plasmids with beneficial alleles, and so if they get a mutation and it works for them, they make a little copy of it and share it with their buddies, even if their buddies are not necessarily bacteria. So they can produce genetic differences that they were not created with by sharing these off to someone new. So that's not really sexual reproduction, but it has aspects of sexual reproduction in it. There are protozoans that can reproduce sexually or asexually, and it may change depending on circumstances. The same thing happens in funguses a fair amount. Alternation of generations is a more strict approach, where in a typical life cycle, from one point in the life cycle to the same point again, you go through both types of reproduction. You go through an asexual stage and a sexual stage. So you get the benefits of copying and the benefits of mixing and matching the genes, of diversifying. And that shows up in 3 different circumstances in living things. Organisms that survive by living in other organisms have huge impediments to being successful. Is that they need to be able to survive in a host long enough to create a lot of offspring, and then the offspring have to be able to find and get into the next host. The assumption is they dip into the advantages of both types of reproduction by, if they manage to get into, and very often this is in separate hosts, they get into a host and then they make copies, right? We were successful, let's make copies of success. They get into a host and then they recombine, often through mating, but sometimes by themselves. But again, we were successful, let's mix and match genes, let's match up genes of successful forms and maybe increase our odds down the road. So you get that in parasites, where it's kind of best of both worlds kind of approach. When organisms move from a watery and aquatic environment onto the land, lots of challenges to living on land. One of the big ones and one of the hardest ones to solve was they were dependent on swimming sperm in order to do their sexual stage. And what the early plants on land did, the mosses and the ferns, is that they went through an alternation of generations where the sexual stage could be produced in a form where what water was available, rain and dew, gave the sperm something to swim through. So it may have been a little bit on a moss or a tiny little plant near to the surface in ferns, but the sperm were able to swim through available water to get to the egg cells. Eventually, the sperm were packaged in pollen, and that gave it much better travel capabilities. But in order to spread on land, they produced an asexual form that made spores, and the spores were little cased, asexually produced packages, and the spores hopefully landed someplace that it was useful to grow into the next moss or the next fern. And they didn't, but actually they were going to grow into the sexual stage. And the sexual stage was going to give rise to the asexual stage. The asexual stage was going to give rise to the spores. The spores were going to spread, start with the sexual stage, and then, go alternate. So they kept the advantages of diversification for sexual, but they used asexual as a way of spreading when water wasn't going to be immediately available. In some animals that are basically non-mobile, they're set in one spot. Some of them, in order to spread across fairly large swaths of ocean, developed alternation of generations where the sessile form, like a coral, is spreading asexually. A large mass of coral is all genetically identical. It's made clones of itself spreading through. But they can produce genetic copies that don't look like coral. They're little jellyfish. And the jellyfish can get up into the plankton layer and spread through the currents. And islands are always rising up from the bottom of the ocean. Volcanic activity pushes stuff closer to the surface. It becomes an environment that a coral can colonize, but it needs to get there. So the jellyfish spread, they get together, they mate, they produce larvae. The larvae swim down. Most of them, there's no place for them when they swim down, but some of them are capable of swimming onto a newly risen seamount. It's not quite crested as an island, and a new coral can grow there. So here the sessile animals have a mobile form, a sexual form that is capable of mating, finding and mating, but also dispersing. Sorry, I was having trouble coming up with the word. We've talked about this, is that homologies, similar structures. For a while, people would parse out the sequence of amino acids and proteins, and they could use that to find connections that you couldn't necessarily connect otherwise. Sea snakes were a mystery for a really long time as to what land snakes they were mostly related to, more closely related to. And one of the early attempts to do that took the venom, which is a protein-based venom, and worked out this amino acid sequence and looked for a similar sequence in land-based snakes. And if I remember right, is they found the connection mostly to kraits, which I think are related to cobras too. But DNA is also a sequential molecule. It can also be compared. The sequences can be compared for homology. And the same way the homology in bones, a bat's wing doesn't look identical to a human hand, but it's got a lot of the same structural elements. You can do that for the sequence of DNA as well. And that's mostly what's done now in classification comparisons, is that they pick select stretches of DNA, usually not code sequences. Because the problem with code sequences is that evolution can drive changes in alleles depending on changes in circumstances. So you can't trust that they've just been changing at a regular rate. You look at stretches that are non-coding and you assume mutations in there just occur randomly at a particular rate, right? Uniformitarianism. We can kind of figure out the rate now. We compare sequences and assume that any differences between the sequence over here in this species and the sequence over here in this species, the number of changes are like ticking clocks. Change, 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 back to a shared ancestor. Now, like any uniformitarian approach, it assumes that mutation rates are steady across a broad swath of time. I mean, what else are you going to do? Is it you don't really know what it did in the past, but we do know that the environment for mutations has not always been the same. That any time that the Earth's magnetic field gets a little dicey, and it does actually turn off and swap over occasionally, you're going to have generated mutation rates from cosmic radiation that are much higher for short periods of time. But who knows? We don't really know what that effect is, so we kind of ignore it. It does often wind up where the bone people, the paleontologists, and the genetics people really don't agree on where a divergence would have happened, because they put it at different points in the past. That, I think, is a good place to stop for this. Let me see what else I'm going to do. Yeah. Now, the next thing we did a bit in the first semester. Now we're just going to come back around and look at it more from an allele standpoint.